Welcome to AP Biology. This video is on the origin of life. Life first originated on Earth about 3.5 to 4 billion years ago. The first living organisms to exist were prokaryotic. The major episodes in the history of life are going to coincide with environmental events taking place on the Earth at that time. The first prokaryotic organisms evolved in structures called stromatolites. We'll see a picture of a stromatolite in a second. Here we see what the Earth's surface looked like about 4.5 billion years ago. It looked very different than it does today. The volcanoes on Earth were constantly erupting, and they were releasing gases into the atmosphere like carbon monoxide, carbon dioxide, hydrogen, nitrogen, and water. The atmosphere itself was very volatile. There were constant electrical storms, and lightning was always present in the atmosphere. The water would condense in the atmosphere and fall back down to the earth, forming the primordial seas. Where the seas met the land in these structures called stromatolites, this is where the first living organisms would evolve. The operant hypothesis describes the abiotic synthesis of organic monomers. In order for prokaryotic organisms to evolve, we first have to begin by taking all the molecules that were present on Earth and forming the basic organic monomers, monomers like amino acids, nucleotides, fatty acids. An experiment called the Miller and Urey experiment demonstrated how these first organic monomers could have been created abiotically in the atmosphere. The apparatus simulated conditions on early Earth. They boiled water, and that boiled water released water vapor into the atmosphere. They placed into their simulated atmosphere all the gases that were present. Also in the atmosphere, they generated electricity that would have been found in the atmosphere at that time. The water vapor would condense and fall back down to the earth. What they found is that the electricity in the atmosphere fueled chemical reactions between these molecules. You can see that all the important atoms necessary for monomers were found. You have carbon, you have hydrogen, you have nitrogen, you have oxygen. The water condensed, fell back down to the earth, and formed the primordial seas. Miller and Urey took a sample of the primordial seas and found that it contained basic organic monomers like amino acids. These seas that had all these organic monomers in them, the Miller and Urey referred to as being the organic soup. Some scientists believe that the first organic monomers were not created in the atmosphere, that instead they are created deep in the ocean by the hydrothermal vents. All the components necessary to create the first monomers were also present there. From these basic organic monomers, we now need to create polymers. Polymers like proteins, nucleic acids, triglycerides. We know that where the primordial seas met the land, we can find sand, clay, and rock. The sand, clay, and rock were all charged, so that water would be attracted to it, would stick to it, and that would cause the water to be removed. Or in other words, abiotic dehydration synthesis was taking place. And that dehydration synthesis formed bonds between the monomers, creating polymers. Some of the first polymers to exist were polymers like protenoids and micelles. A protenoid is an abiotically produced polypeptide. A micelle is a self-assembled lipid macromolecule. On the right you see a picture of a micelle, and it's made up of phospholipids. We know that phospholipids have a polar head and they have two nonpolar fatty acid tails. All the nonpolar fatty acid tails would face each other, and the polar phosphate heads would face the polar water. These phospholipids can just as easily form a phospholipid bilayer, where the fatty acid tails would face each other and the polar phosphate heads would face the water on either side of the phospholipid bilayer. We know that this phospholipid bilayer would eventually form the cell membrane. When these early cell membranes were formed, some other monomers might have been trapped inside, or some polymers could have been trapped inside. And what would form are these early structures called protobionts or protocells. They weren't true cells because they didn't have the ability to reproduce. However, they were the early precursor to cells. In order to create a cell, the cell needs the ability to reproduce. Scientists believe the first genetic information was probably RNA and not DNA. They believe it's RNA because RNA is much more simple. It's single-stranded, and it's a lot easier for it to replicate. The RNA can replicate autocatalytically simply using the base pairing rules. These early protobionts with self-replicating catalytic RNA would have been more effective at using resources and would have increased in number through natural selection. According to the heterotroph hypothesis, the first living organisms were heterotrophic prokaryotic bacteria. To be a heterotroph means that you need to consume food for energy. And scientists believe that these heterotrophic bacteria fed on the organic soup. From the heterotrophic prokaryotic bacteria evolved autotrophic bacteria. These autotrophic bacteria use the process of photosynthesis to make their own food. Cyanobacteria are an example of autotrophic bacteria that existed at this time. Photosynthesis released oxygen into the environment. This allowed the ozone layer to form, which is going to protect these organisms against UV damage. When oxygen was released into the environment, this also opened up a new way to produce energy, a new method that would produce far more energy that would allow life to become more complex. Aerobic respiration. The endosymbiotic theory explains the evolution of eukaryotic cells from their prokaryotic ancestors. In particular, it explains the formation of mitochondria and chloroplasts. The endosymbiotic theory states that eukaryotic cells evolved as the result of one prokaryotic cell engulfing another prokaryotic cell. These two cells formed a close and permanent relationship. Both benefited from their relationship. It began with a large cell engulfing a smaller cell. That smaller cell was extremely efficient at producing ATP. It contained electron transport chains. The small cell benefited from the large cell because it was given a constant supply of food. The relationship between these two cells was so beneficial that the smaller cells became permanent inhabitants within the larger cell. You can also see how the first membrane-bound organelles could have formed. Membrane-bound organelles could have formed by having the cell membrane fold inward. This could have given rise to organelles like the nucleus, endoplasmic reticulum, Golgi apparatus, etc. Having membrane-bound organelles is one of the characteristics of a eukaryotic cell. Now that these cells contained mitochondria that could produce lots of energy, eukaryotic cells could become more complex over time. There's a lot of evidence that supports the endosymbiotic theory that mitochondria and chloroplasts were once their own separate prokaryotic organisms. Mitochondria and chloroplasts both have their own circular DNA, both have their own ribosomes, both reproduce independently of the cells that they inhabit. This information proves the idea that mitochondria and chloroplasts were once their own separate prokaryotic organisms. The first multicellular organisms were probably colonies. Here you see a colony of algae. Algae are protists. At the beginning, each protist within the colony would be performing every single function that it needs to survive. After a period of time, the colony would start dividing up the labor. Cells in one region would perform particular functions. Cells in another region would perform other functions. After a period of time, the entire colony would be functioning as one multicellular organism. Fast forward millions of years, during the Cambrian period, in particular the first 20 years of the Cambrian period, life saw the Cambrian Explosion. The Cambrian Explosion is when most major groups of animals first evolved. Plants, fungi, and animals, they colonized the land about 500 million years ago. There are many different methods of classifying the life that we have on Earth today. Here we see different classification systems. The major system that we use today is the three-domain system. Domain 1 is bacteria. These would be all the bacteria that we think of as being germs. Domain 2 is archaebacteria. Archaebacteria are the bacteria found deep in the ocean by the hydrothermal vents. And the third domain is eukarya, all eukaryotic organisms. All eukaryotic organisms can further be classified into 4 kingdoms. There's one big group right here would be protista, protists, organisms like algae and protozoans, plants, fungi, and animals. These kingdoms can then be further broken down. As a final note, I want to stress the importance of mass extinctions in the diversity of life. People think of mass extinctions as typically being bad. They're an important part of life because they open up new adaptive zones and allow for the formation of new species. You can see through the history of life there's been a bunch of mass extinctions. You see the Permian extinction, the Cretaceous extinction. These extinctions opened up new adaptive zones which allowed new species to evolve. So if you look at life over time, you can see that life has experienced an increase in biodiversity.